So when we talked about the natural killer cell synapse, uh, we mentioned kind of all the processes that are involved in that. This is analogous, I think of. It may be homologous because they obviously have a similar cellular origin. But anyways, there's two parts of this. There's the peripheral supramolecular activation complex, abbreviated here as the PSMAC because it's in the periphery. And this is just a seal to make sure that we have everything that stays tight and stays in place. And then there's the central supramolecular activation complex, which is the actual part of activation, containing, the T cell receptor, the co-receptor interactions, and then the co-stimulatory signal, which we'll talk about later. Now, the binding of the antigen by the T cell receptor causes the other T cell receptors and co-receptors as well to cluster on the membrane, initiating intracellular signaling. I just like to remember this as being really similar to the natural killer cell synapse, where once we have one there, all these other molecules are going to start to migrate to that area of contact. And there's a lot of physical processes involved in initiating the signaling that's involved in this. And I love this picture here because it shows just how complicated it is. So here we see in purple the T cell receptor, and then in blue is the T cell co-receptor. In this context, is showing the CD4 molecule because, it's MHC class 2. That's what we're talking about here. And then we have all this stuff downhill from it, all these different signals. It's this huge, massive cluster of signaling back and forth between the two. And this is just the ones that we know of right now, but the only ones that I think are important in terms of studying for this class at least is obviously the T cell CD4, the CD28 interaction that it has with B7, and then lastly the CD40 interaction is really, really, really important. All this other stuff in between here that's really fun and it's interesting, but I don't have time to talk about it in grave detail, and so we're going to stick with the stuff that we at least know of in terms of its importance, in terms of its pertinence. Go over this. I wanted to talk briefly about the signaling cascade, and I'll put a link in this to, or maybe something up here, to my biochemistry videos where I talk about the roles that tyrosine kinase type receptors can have in terms of like signal transduction pathways and mediator proteins and all of that stuff, because I think that's what we're going to talk about here, and at least in a little bit, not in nearly as much depth as we do when we talked about biochemistry. This signaling cascade here, so let's just kind of begin it. So we start off with the classical T cell receptor interacting with both the MHC molecule and the peptide. And then the co-receptor is obviously going to interact with the MHC molecule in this context, since they're showing it as CD4, but it could just as easily be CD8 if we wanted to, but this is more specific to that type of a process here. And so attached to this is the soluble kinase known as LCK. LCK, whenever this binding here happens, is going to start to phosphorylate all of these regions called ITAMs. Now, what an ITAM stands for is an immune receptor tyrosine-based activation motif. And this is just an area where, in this context, the LCK, which is just a soluble tyrosine kinase, is going to go, and it's going to phosphorylate the specific regions, the specific motifs in the CD3 complex. Okay, and so once that's phosphorylated, the compound ZAP70 is going to be bound to it and kind of held in place in order for LCK to come in, phosphorylated, and then the phosphorylation of ZAP70 has other events downstream. We'll go in more depth about this stuff later, but understand that it's this long series of phosphorylation of signaling cascades that are happening. And this is just, mind you, this is just this part right here. We haven't even talked about all this stuff here. So one of the things that you're probably going to notice is that the decision to activate T cells is extremely precise and complex. And the more complexity we have in our axes, the more complexity we have in a signaling cascade, the better we can fine-tune it and the better we can control it. And that's why this is so long and complex. That, and you got to think about where this evolved from. But anyways, so this is a picture here, just kind of, hopefully you can see it on the screen. It is a little bit small, but the naive T cell interacting with the dendritic cell specific antigen. This is going to activate ZAP70. I'm going to map this, hold this stuff out later, so don't worry about it. This is going to ultimately, with the help of other compounds, result in the activation of phospholipase C, and then phospholipase C, in case you don't remember that, is going to, through the result of the cleavage there, end up producing the second messengers, DAG and IP3, which, DAG does its thing and activates, in this context, protein kinase. I think that's theta? I'm not sure. I'm not Greek. I don't know that. But anyways, it activates the ROS protein. Remember that we talked about ROS and SOS and that axes there. Again, I'll put a link to my biochemistry video that kind of talks about this in more depth. There's inositol 3-phosphate, which always results in the release of calcium, and then calcium as a second messenger is going to result in the activation, with the help of others, of calcineurin, which is going to activate something known as INFAD, which is called nuclear factor of activated T cells. But one of the things I just want you to really think about is that all of these processes here ends up resulting in the production of transcription factors that are going to result in, ultimately, when they reach their target sites, cell division, proliferation, and differentiation. Okay, so early activation events are predominantly involving tyrosine kinases. Tyrosine kinases really are always involved in cell division and then cell maturation and things like that. And then later events, though, are going to involve protein kinase C. Notice that we're talking about kind of later. So like with earlier events, we talked about the whole phosphorylation and signaling cascade. But later on, we're going to talk about protein kinase C, and then a new type of interleukin, or a new type of cytokine that we haven't talked about, known as interleukin 2, which is, it's dubbed as the T cell growth factor, right? Because that's what I think you can apply from that name what it does. Okay, so interleukin 2, as we talked about later, is the T cell receptor binding to an MHC peptide complex induces, so notice that it's this, just T cell receptor MHC. This induces the synthesis of interleukin 2, wow, and CD25, which is the alpha chain of the receptor for it. So one of the things we may want to notice that this is a autocrine type of function. It's going to make cytokines that are not only going to activate on others, but also mostly activate on themselves, right? And producing a very high affinity for it. So here we see the naive T cell with the gamma and beta. That's always there, that's always present. But once it becomes activated, usually through interaction with the TCR MHC peptide complex, it's going to start to produce alpha. And then alpha is the one that gives it, one, a higher affinity, but two, plays a larger role in the whole cascade of events that result in the signaling processes. And then and kind of has this, logarithmic effect. So co-stimulation through the CD28 binding to a B7, which is something that we haven't really talked about in too much depth, but just understand that when we talked about that this right here is an extremely crucial part. I'll mention it in further detail in my map, but it's also one of those things that I showed on the past page that's really important, that's really unique to humans especially. This is going to increase the transcription of interleukin 2 gene, and then threefold, and it also stabilizes the interleukin 2 messenger RNA, causing an increase in the production of it. However, my textbook doesn't give details into the mechanism of how it stabilizes the messenger RNA, so don't ask me because I couldn't tell you. But if you know, please let me know. I'd love to hear about that. Proliferation and differentiation of activated naive T cells are driven by the cytokine interleukin 2. Its nickname is the T cell growth factor for a reason, right? And so this generates an autocrine feedback loop resulting in clonal proliferation, right? 2 becomes 4, 4 becomes, it has this logarithmic kind of growth for it. And this, so the thing that we really want to make sure of, earlier when we talked about the complexity of it, but 2, think about how much effort we want to put into controlling the growth of our lymphocytes and of all cells of the immune system really, but T cells as well, because if you have uncontrolled growth of especially things like this, that's extremely bad, because lymphatic cells, or all immune cells really, have telomerase activity, which if I'm assuming you've taken general biology, you know what that means. And 2, they have extremely they have a really longevity. They can live for a really long time. So if that's something that becomes cancer, that's going to be a really bad form of cancer. But 2, if it grows, there's not a lot of intrinsic mechanisms that are designed to stop it. So we must be very careful with these signaling that we're doing. And that's that's why we have this. The CTLA4 inhibits the effects of all this proliferation. It's the exact opposite of that. So it's going to kind of put the brakes on it. So it prevents uncontrolled cellular expansion so that it doesn't just grow and grow and grow and grow. So the mature dendritic cell, once it's been activated by an antigen, is going to be constitutively expressing CD40 and then B7. Switch colors to that. The naive T cell, notice what we're talking about naive T cells here, are going to be constitutively expressing CD28. And then ultimately the interaction between the T cell receptor with its MHC complex, the coreceptor, and then the CD28, all this binding is going to induce the expression of the CD40 ligand from obviously on the T cells, right? So the T cells are expressing CD40 ligand, and that's going to interact with the CD40 receptors on the dendritic cells, and this is going to upregulate the expression of the co-stimulator B7 on the dendritic cells. And so what you may be thinking is, well, this is about signaling involved in proliferation and differentiation, and it has a positive interaction to where 2 becomes 4, and then 4 becomes 6, and so on and so forth, right? And so this obviously could very quickly get out of hand, is the point that I'm making. And so yeah, like I had said here earlier, or like I had said previously, that this binding is clearly, it's very necessary, essential. It can't happen if we don't have this. If we don't have this interaction, the naive T cells will not become T cells. They'll become energetic and they won't do anything. But once they're activated, the T cell is going to start to express something known as CTLA4 that's going to bind to B7 with a 20 times greater affinity, and CTLA4 actually is antagonistic to the signaling processes. So this is how we can kind of shut things down and kind of put the brakes on this massive response of cellular proliferation and cell growth, because for every one CTLA molecule, we'd have to have 20 more of the CD28 binding. And then also something that we're not listing here, though, is that once the T cells become activated, they stop expressing CD28. So that's pretty cool news. And this is something from my straight from my textbook. I just took a picture of it, and I just wanted to read it with you because I think it's, one, crazy to me that this was discovered, but two, it really highlights the important role that the co-stimulatory signal plays, not just in humans, but in terms of distinctions between us and really closely related relatives. So this is what it says, that the importance of CD28 and T cell activation was tragically illustrated in 2006, not that long ago by my standards, by a phase 1 clinical trial in which 6 healthy volunteers were given a humanized anti-CD28 monoclonal antibody. This type of antibody had been shown to ameliorate autoimmune diseases in a rat model. On binding to the CD28, the antibody mimicked the natural B7 ligand, inducing a signal from CD28 that activated regulatory T cells, which then acted to suppress inflammatory effector T cells in a non-specific fashion. On the basis of these encouraging results in rats, the humanized anti-CD28 antibody was being tested as a possible therapy in humans to reduce T cell-mediated inflammation that characterizes arthritis and many other autoimmune diseases. The phase 1 trial was a necessary preliminary test to determine the effects of the antibody on healthy humans before giving it to the patients. The antibody was never given to the patients, however, because of its catastrophic effects on these healthy volunteers, which was the exact opposite of what was expected. Instead of inducing a suppressive response, the antibody activated an army of effector T cells that produced a storm of cytokines and chemokines, causing systemic, life-threatening inflammation and autoimmunity. So this is basically the way that what would happen if we didn't have that CTLA4. We would die, basically, is one of the things we said. It says all 6 volunteers ended up in a hospital as patients themselves. One of them remained in a coma for 3 weeks after experiencing heart failure, liver failure, and kidney failure, as well as suffering from septicemia, pneumonia, and gangrene. So it didn't even... The overactive, this is just another sign that the overactive immune system is not always a good thing. Such effects had not been observed in the antibody when the antibody was similarly given to healthy monkeys, and many of the consequences emanating from this catastrophe was investigated to explore the biological differences that distinguish human CD28 from its rat and monkey counterparts. So think about that for a second.